The first flash talk of this session is by Qi Yin.
Okay, good afternoon, everyone. It's such an honor to present here today and get valuable comments from all of you. My name is Qi Yin from Liangshan, Southwest China, and now I'm a postdoc in University of Cambridge. We are currently studying the relationship between Enterobacteriaceae and human gut microbiome, as well as the ecological pattern of Enterobacteriaceae colonization success. Now I will share with you the results of our project. Enterobacteriaceae, particularly like Escherichia and the Klebsiella, are a global health concern. The rise of antibiotic resistance of these species leads to increasing morbidity and mortality globally. Thus, they have become the WHO priority pathogen list for discovering novel antibacterial drugs. Luckily, we have plenty of genetic and metabolic resource inside our body. The human gut microbiome contains the highest microbiome diversity and density. However, we have limited knowledge about how Enterobacteriaceae interacts with the rest of gut microbiome. Exploration about this topic might give us novel hints to control Enterobacteriaceae colonization via modulating gut microbiome composition and environmental condition. We analyzed over 25,000 human fecal sample metagenomic data from public database involving 88 studies across 53 countries based on our comprehensive catalog of bacterial genomes previously generated. We used the prevalence and abundance of these species to identify significant species associated with Enterobacteriaceae. Then we use machine learning and network analysis, try to further select target species for functional and metabolic characterization. As for the main variables of our samples include health state, continent, and age group. To obtain robust signal, we divided our samples into 3 data sets: all samples, high-quality information samples, and healthy adult samples, and use different confound variation as mixed effect to perform the statistic analysis. The prevalence of Enterobacteriaceae is highly among Africa samples and infant samples, as well as RA patients. And Escherichia and Klebsiella are the most prevalent general across different health states, age group, and continent. And machine learning based on gut microbiome abundance revealed that the microbiome composition is highly predictive of Enterobacteriaceae colonization status, with AUC ranged from 0.7 to 0.8, and the results are consistent across the 3 different data sets. It's confidential to say that gut microbiome composition could be a biomarker of Enterobacteriaceae colonization prediction. And this is the final genetic tree of the microbiome species significantly associated with Enterobacteriaceae. We combine the signal from abundance and colonization standards. Overall, we obtained over 600 species significant associated with Enterobacteriaceae. The slide was colored by the isolate standards. Blue is for isolates, and green is for max. Over 50% of these species are uncultured, which means we have a lot of information to explore. And the inner circle represents the lineage information of these species. And for the outer circle, we have 3 paired circles represent association with Enterobacteriaceae colonization and abundance in different taxonomic level, E. coli, Klebsiella, and all Enterobacteriaceae. And the color represents the direction of this association. The blue is for negative and the red is for positive. Clear species cluster between negatively or positively colorated species were observed, and the species from Bacteroides have the highest, are enriched in Enterobacteriaceae absence, especially for allostips and Privitella. And we can also see here, there are a clear strain variation inside the same group, which means investigation about the functional genetic differences between these species is worth of further exploration. And last but not least, we perform the functional analysis. The KGG ortholog diversity and the metabolic distance of the species are negatively or positively associated with Enterobacteriaceae reveal that species associated with Enterobacteriaceae colonization are more functionally diverse and metabolically closer. And on the basis of the distribution of COG, species negatively associated with Enterobacteriaceae colonization encodes more functions involving cellular process and signaling. The conclusion of our project so far, Enterobacteriaceae are highly prevalent worldwide, and the microbiome composition is highly predictive of Enterobacteriaceae colonization status. Bacterial data species are strongest biomarker of Enterobacteriaceae absence. Species co-occurring with Enterobacteriaceae are metabolically similar and more functionally diverse. Thank you, that's all.